Welcome, everyone, to Biosafety Now's 16th space, our second X space. My name is Bryce Nichols. I'm a professor of genetics at Rutgers University and lab director at the Waxman Institute of Microbiology, and I am one of the co-founders of Biosafety Now. Biosafety Now is a U.S.-based nonprofit organization launched this past February, led by experts in biomedicine, mathematics, public health, public policy, law, social science, and public advocacy. Our core mission is to work for a future where scientific research on pathogens supports human life without threatening it, and public trust in science is restored. You can learn more about our mission by visiting our website at biosafetynow.org. I also want to give a disclaimer at the beginning of tonight's space that the views of individuals expressed on these spaces do not necessarily reflect the views of Biosafety Now. Now, tonight I'm going to be joined, I guess, in a little bit later by another one of Biosafety Now's co-founders, Justin Kinney. He's a professor of quantitative biology at Cold Harbor, and he is on a train right now. Unfortunately, I apologize because I'm sad to say that a new member of Biosafety Now that I had said would be joining us, Megan Gafford, will be unable to join us this evening because she's recovering from an illness that's caused her to lose her voice. I look forward to introducing everyone to her, and I hope she can join us on our next Twitter space because we're very excited that she's joined us, and I can't wait for everybody to meet her. I would now like to introduce our featured guest this evening, Dr. Jonathan Latham, co-founder and executive director of the Bioscience Resource Project, an organization that publishes independent science news. They're a nonprofit organization just like we are, so you can go to bioscienceresource.org, check that out, and donate if you would like to support that. I'd also point out that Dr. Latham holds a master's degree in crop genetics and a PhD in virology, which is something we'll talk about a lot today. I actually wanted to start tonight talking about an article that you wrote, I guess it was in June or July, titled The Great Raccoon Dog Mystery. Now, I want to go back to something you mentioned in the bio on your website that I didn't point out, but I'm going to use this to introduce the subject covered in The Great Raccoon Dog Mystery. You have this statement. It says, Dr. Latham talks frequently at international events and scientific and regulatory conferences on the research conducted by the project. Now, what we're going to begin tonight talking about is, in fact, you visiting conferences and being censored and unable to speak about your research. So do you want to give us a little bit of background on that essay, The Great Raccoon Dog Mystery, and what it's about?
Well, so what happened is that it's difficult to get questions answered when and Ed Holmes doesn't answer your questions, Andrew Rambo doesn't answer your questions, and there are a lot of there's a kind of asymmetry in the discussion over COVID origins. And so, people who are proposing zoonosis will only talk almost entirely to what they consider to be friendly media sources, and those friendly media sources are not asking very penetrating questions in the main. And so I thought that I could do that and obtain a little bit of clarity about some of the issues surrounding COVID origins by going to some scientific meetings this spring. So I went to the Pathogens Project meeting that was in Switzerland at the end of April, the 20th of April. It was a 2-day meeting. I went to the European Congress of Virology, which is a 5-day meeting in Gdansk, Poland. And I went to the coronavirus meeting. It's called actually NIDO 23, but it's basically a coronavirus meeting, also in Switzerland, that lasted 5 days. And it turned out the last meeting almost entirely was about SARS-CoV-2. And a huge number of people, especially at the last meeting, who are, have been in the thick of the discussions of COVID origins, people like Ed Holmes and Bob Gary, were all there at this meeting, and it turned out to be the perfect venue for me doing what I wanted to do, which was firstly learn what people were thinking, and provide some information, and maybe explore some new questions too.
So, Jonathan, I want to back up because I want to go through all three, because in your article you sort of begin with the pathogens project. So could we start there? In fact, the pathogens project, your participation in that was the first time I actually became aware of you, for two reasons, I guess. One, because of your infamous handouts that you brought to that meeting. And then the second was you gave a very interesting speech, met with some dissatisfaction by the attendees, or perhaps that was just my perception on the looking at the watching it on the web, where you mentioned Richard Ebright as somebody that you really trusted to sort of try to regulate dangerous pathogens research moving forward. So do you want to set the scene for us about that pathogens project meeting? I guess maybe also give the listeners some background on what the purpose of that meeting was.
Well, the pathogens project meeting is a meeting that was first mooted, to my understanding, by Alina Chan. She wanted to have a discussion about the origins of COVID, and it didn't turn out that the origins of COVID was really a topic of that meeting, but nevertheless, Alina is one of the members. So is Jesse Bloom. There are David Relman. There are a bunch of people who are members of the Pathogen Project. Mark Lipsitz, probably names familiar to many people listening here. And I was, I had some reservations about the nature of the Pathogens Project. I wanted to know if it was, if they were, in a sense, sincere in their efforts to organize change, to make lab, I'm interested in making lab research, it's basically the same as Biosafety Now. I'm interested in making lab research safer, and if necessary, banning certain kinds of research. And I felt that, the history of social movements is that many times there are, radical institutions start up that want genuine change, and then what happens is some institution pops up. A classic example will be Science for the People. In the 1970s, Science for the People started as a radical sort of socialist voice on speaking about science. And soon after that started, the Union of Concerned Scientists started up, which basically assumed the same role, but in a kind of a much more diluted form. And so this is a recurring pattern, and the impression that is always gained is that the sort of lukewarm version of these institutions is successful, and the radical version is not successful. And the lukewarm institution basically is getting in the way of actual genuine change. And so I wanted to know, is the Pathogens Project really sincere in their desire to have research made safer? And I didn't know the answer to that question. And so, the sort of chief goal for me was, if you meet these people, you get a much clearer sense of whether they are genuine in their concerns, and how determined they are to get the job done, if you like.
So and that's a very interesting comment. You put it very politely, actually. I think, some people, I guess, call that controlled opposition. Some might refer to it that way. Mm-hmm. But there is this question of how sincere people are about what they say publicly versus what they really believe privately. And certainly, we've seen with the proximal origin case where it's a dramatic difference. But with respect to regulating pathogen research, I guess, what was your takeaway from that meeting? What conclusion did you come to about the sincerity of those efforts?
The bottom line is, I'm not going to say that everybody there is sincere or everybody there is not sincere, but I wasn't fully reassured. There were a lot of people who were at the meeting who I would say were definitely not sincere and how much involved they are in the Pathogens Project. I mean, one of the Swiss researcher that I'd never heard of before ended up being a big part of the lab the story of the meetings that I went to, because he was at every single meeting that I went to, and so he was on the platform of the pathogens project, and he made some statements that clearly, they seem to me utterly unreasonable and not the kind of things that a person would make who really was sincerely concerned about lab research. And he's a vaccine researcher. He's intimately involved in all this, a lot of vaccine research, and his personal take on this. For example, one of the statements he made at the meeting was the Pathogens Project meeting was that essentially it's about whether people follow the rules, and in his country, people always, which is Switzerland, people always follow the rules. And so, I don't agree with the premise. I don't think it necessarily follows that if you follow the rules, there are no lab leaks or escapes or whatever. But also, it seemed to me ridiculous to say that in some countries people obey the rules and some people, and in other countries they don't. It's clear that in the United States, for example, people don't always follow the rules. That's, that's something that's just become so obvious in the course of this discussion. So there's certain comments that various people made did not speak to me positively about their sincerity or understanding of what the deeper issues are. And if you want to make change in this kind of a space where there are powerful pharmaceutical interests, there are powerful military interests, there are powerful research interests and commercial research interests, you really have to have your wits about you and be really determined and clear about what you want to do. And I didn't really get that feeling from the Pathogens Project as a whole.
There's a long history of, there's at least a decades-long history of trying to rein in risky research, like by the Cambridge Working Group, and through these funding pauses and different, regulatory frameworks. And I think, largely, at least, in Biosafety Now, we think that it's been a catastrophic failure. And my biggest concern with the Pathogens Project is that it's just trying to kind of repeat that. I was wondering if, A, they talked about this at all, and B, what your take on that.
Very briefly, Mark Lipsitz discussed a few of the lessons of, and a few of the positives and the negatives, but of the history that you're talking about. But for example, that would have been a great topic to actually focus on. But it really wasn't done. There was no sense of a learning process. The feeling was that there were a lot of people in the room who really were not familiar with the biosafety debate. And there were a lot of people who hadn't, really deeply thought about those lessons. There was a lot of people, I couldn't work out why they were there. Do you know what I mean? And it felt like they were there as ornaments or something like that, and as prestigious people or as people who might have an in with Fort Detrick or something, but they weren't evidencing a clear vision of improving biosafety or improving science or making pathogen research safer. They were, many of them were simply defending the status quo. And to me, inviting people whose main contributions is to defend the status quo is not evidence of sincerity.
So you mentioned that that there wasn't a lot of talk about the origins of COVID. Was it like just something they weren't interested in? Was it just not considered polite to do that in the gathered company?
Yeah, so I would say that the impression I got was that it was not considered polite. Do you know what I mean? There were too many people in the room who would be offended by a genuine discussion of, whether the WIV was telling the truth and so on and so forth. So that was the feeling, and it's not true that no one talked about it, because George Gao taught at that meeting, and he did discuss the origins of the virus, but it was extremely superficial. It was not really in depth at all. And he talked, and the sense I got from his talk was, it was a sort of stand-up comedy routine more than a serious attempt to address the science. And I felt like he was someone who, really we could learn a lot from if he could tell what he knew, but he wasn't telling what he knew. And so to me, it seemed more like a charm offensive and a stand-up comedy routine with very little substance involved in it when George Gao spoke. And yet he comes all the way over from China, or at least from Britain, I think he was previously, to talk about that. And it just seemed really empty.
So I guess I wanted to interject something before we got cut off, which was that one thing that, to be fair about the Pathogens Project is that they've yet to release their report, right? So when we had Alina Chan on a while ago, she mentioned that she's been working on that report. And I know, for one, I know that she's very sincere in her efforts and her participation in that. And I think there are a lot of people involved, and I guess it's unclear what their motivations are. But as I said to you, Jonathan, on the phone, I said I'm a little hesitant before we see the report to be too critical about it. In part, just out of respect
Yeah,
for
that's fair.
Alina. Personally, though, I had a lot of concerns that you did. I would like to know, in terms of the environment itself, who were you interacting with at the meeting? Because we weren't at the meeting, but we saw the speeches and those chats they had on the stage. But in the lunches or in the breaks, who were you speaking with, and what were those conversations like?
Well, those conversations, it was clear that some people didn't want to talk to me. It was clear that some people who were there knew absolutely nothing about the subject. There was one lady that I met from Jordan, and she knew nothing about any of the evidence, pro and con, lab leaks. It's entirely unclear why she was there. There were other people who I talked to a social scientist, and I talked to him about some of the lab leak issues, and he gave us all these signals of being not comfortable talking about these issues. And I was trying to think, well, why are you here if you don't either know anything about the concerns over a lab leak, or you're simply uncomfortable talking about these issues? I wasn't even trying to get this fellow to express any difficult opinions, but I also, I feel like it's useful. The reason why I go to meetings like this is because, I want to challenge these people a little bit. The idea is not to just put them in their comfort zone and talk about the weather. The point is to initiate a conversation about something that could be interpreted as a little bit difficult. And some people will just meet you and they will just want to frankly discuss the question as a simple question of science and so forth. And other people are really uncomfortable doing that because, they think about how it would be to talk about it at their institution. They're thinking about all these other issues, how it will affect their career, how will it affect what their friends think, what happens if they're seen talking to me, and so on and so forth. But those people, there are many such people, but those people are not of any use in a social struggle like this because they're not concerned primarily with the public good or finding out the truth. They're concerned primarily with their careers and where the money's going to come from and so on and so forth. So you get a sense of who these people are and what they think about. And I wouldn't say that I talked to any of the leading people. I talked very briefly to David Relman and a few other people. But, for the most part, those people were either very busy or they didn't want to talk to me.
I will say, I guess, to clarify something about this conference. One is that we, let's see, Jesse Bloom, invited several members of Biosafety Now to participate in it. So there was some choice there in terms of, I think due to travel constraints for many of us, that was one of the reasons that we didn't attend. I did find, though, that there were some notable omissions in terms of the journalists that were platformed there. I had mentioned before that it was disappointing that Emily Kopp was not put on a stage there to talk. I think that was very notable, a huge loss for this issue to not have her there. But I think, as you alluded to, the protection of careers and sort of group status is something that is hampered really addressing this issue. And it's unfortunate. One might understand that prior to COVID, but after COVID, it's simply inexcusable. Um, and I know that maybe perhaps at some point, Sam Husseini might want to talk about that later on. I don't know. I don't want to. He had some thoughts about that when he was on the space a few months ago. I did want to now go to your second meeting.
Bryce, before you go on, can I just say, like, I'm generally quite supportive of the Pathogens Project, and I have immense respect both for Alina Chan and Jesse Bloom, and I think Alina Chan, it should be noted, really has taken a lot of major risks with her own career to raise the subject of COVID origins. But I have been concerned that when you get a group together, that everything will just get watered down. And, people not wanting to bring up difficult subjects will, can, cause like the core topic to just be avoided. So anyway, but I think, we should withhold judgment until the Pathogens Project report.
I think, as you said, different people have different motivations. And I do also have a lot of respect for people that participated, some of the people that participated in that. And I really would love for, when that report comes out, for members of the task force to participate in a Twitter space to actually discuss this and engage with members of the public. So that
that would be great.
I would hope that Alina would certainly do that, and I would encourage. I've invited Jesse to come on, and Mark Lipsitch as well would be nice if he would come on, but we'll see. So can we now talk about the first scientific conference that you went to? I went out in the hallway. So Jonathan, what was the second conference you went to? Was the European
Congress of Virology?
Okay. So I know I'm very excited to talk about the NIDO conference, but the second conference is notable. I guess you want to. This was a general virology conference, right?
Yes, it was. Yeah. So there was HIV research. There was all shapes and forms, plant research.
So this is now early May in Poland. 1,000 people there, and you actually. So in that essay that I alluded to earlier, you had this notable exchange about the sensitivity of raccoon dogs to SARS-CoV-2. If you want to talk about that and how that relates to your experience at the European Congress of Virology.
Yeah, for sure. So one of the things that that was pointed out to me by the Twitter user Dao Yu a long time ago was that the raccoon dogs are arguably the strongest candidate for animals to have brought the virus to the Huanan market, or to Wuhan at least. And but what's interesting about there's only one experiment that's ever been done to test the sensitivity or transmission of SARS-2 in raccoon dogs, and the results are quite ambiguous. And what Dao Yu pointed out is that the virus version that was used was the D614 mutation and not the I've said it backwards. It was the G mutation and not the D mutation at position 614 of the spike protein. And this mutant is known to be much more infectious in people. It's known to be much more infectious in all the animal species that have been tested. It's known to be more infectious in airway cells, for example, or more viable. It outcompetes the D. So G outcompetes D, and the G mutation basically displaced the D mutation quite early in the pandemic. It was first spotted in January, and within a few months was essentially the only game in town. And so the fact that essentially what these raccoon dog researchers had done, and this is done at an institute in Germany, what they run by this a professor called Martin Baer. They had what the researchers had done is not test the origin virus, the D. They tested 614G, the more infectious version. So they'd essentially done an experiment with the wrong virus, and a virus which is known to be much more infectious than the origin. So the question then becomes, are raccoon dogs in fact capable of being infected even by, let alone transmitting, the D, the origin strain? And so this clear kind of misstep in the research was really a focus of my attention at this meeting. So I really wanted to find out. And when I found out that Martin Baer was speaking, he was the supervisor of this research, then I wanted to ask, why was it that they did not test the origin strain? And his answer was that they didn't know which was the origin strain, whether it was D or G. And my my thinking about this is, even he has said in scientific papers that D came before G. Everyone knows that D came before G. The answer was ridiculous. But in a sense, for me, it was a teaching question. I wanted to show the audience that they had not done the right experiment, and that they knew that they'd not done the right experiment, which is what I suspected in the first place. And that's what his answer, in my opinion, proved, that such a weak answer to such an important question implies that they really knew that they hadn't done the right experiment. And so then the question becomes, why has no one else done this experiment? The whole of the world of virology wants to know whether raccoon dogs can be infected by the origin strain, and no one's done the experiment. The way that I thought about it is, he's shown himself up to be a phony. He's not trying to do real research. He's simply trying to cover up for the lab leak.
Well, I just wanted to comment that I completely agree with Jonathan. It's insane that we don't even know if raccoon dogs are susceptible to the virus that emerged in Wuhan. It's insane. I mean, SARS-CoV-2 is the most studied virus in the world, right? And in history. And we don't know this basic fact, which is key to figuring out where this came from. So I agree. I think I think the real question is, why aren't other people doing these experiments? And I kind of think the answer is probably they don't want to know the answer.
So, with respect to not wanting to know the answer, that's a good transition to the NIDO conference. So, if you want to, so this is the one that I just found to be the most incredible exchange. Well, the most incredible thread I've ever read on Twitter, or among them, was your description of what happened to you when you tried to attend the NIDO. Now, is it NIDO or NIDO? I don't know. I heard you say NIDO.
I think Nito is what I heard. I say Stuart Neal is on the line. I think he probably knows what the correct pronunciation is. But I heard Nito.
That's not Stuart Neal. That's Stuart Newman.
Oh, Stuart Newman. Okay, I just see Stuart N. Yep.
Well, if Stuart Neal was in here, that would make some news, I think. But and again, they have open invitations. People are not blocked from this account, so everybody can access the space. But so that conference, to set the stage, do you want to give an overview of who was in attendance at that conference, and also, I guess, who was speaking? Because a very notable speaker did it remotely.
So the featured headline, if you like, speakers at that meeting included Ralph Barrick, Zhengli Shi, Ed Holmes, and a host of other people who've been very forthright in defending the zoonotic origin of SARS-2. So, and it felt to me actually like there was a convention of such people. I mean, Bob Gary was there, who has no interest in coronaviruses, research interest in coronaviruses, is nevertheless at the NIDO meeting. Joel Wertheim was there. Michael Warraby was there. It felt like Stuart Neal was there. A whole convention of people who are the defenders of the lab of the zoonosis. And so even though NIDO is the general class of viruses that was being discussed, almost every single talk was about COVID-2. COVID-19 rather.
And you said that so Zhijing Li appeared
Yeah.
by remote, and I believe you. So your mission when you went there was to finally get a chance to ask direct questions to some of the scientists that have been making claims that you disagree with or find to be unpersuasive, which of
Yeah.
course is what scientific meetings should be about. And when you got there, do you want to describe what happened initially? Because this gets to those handouts that I mentioned, I guess, two spaces back. The first space that collapsed, I mentioned something about, I said, your infamous handouts. Can you explain what you brought to the meeting?
Well. So I had two articles that I decided to bring. So one was our work on Ebola virus. So we think there's a strong possibility that the Ebola 2014 outbreak was from a lab. And the second one was our theory of the Mojiang miners' origin of COVID-19. So I bought these two, quite large articles. I spent 1,000 Swiss francs printing them out. And when I brought them to the conference, I asked where we could share materials, and I was told to, we can put things at the end of the reception desk. And when I was unpacking them, one of the staff members noticed the title and said one of the titles, and they said, we have to ask permission first whether we can put these out. And to me, I was only asking where to put them and so forth out of a courtesy. I didn't think that they had any reasonable right to turn me down. But I took several hours to get an answer that they assured me would just be there in 1 second. And eventually I was told I was not allowed to share those materials.
And that decision, it turns out, was, I believe, made by that Swiss researcher you mentioned.
Yes, Volker.
Yeah.
I'm blanking on his name. Volker Thiel, I think
Volker
it was.
Thiel.
Yeah.
Yes. So the reason that I noted that name is, of course, so he was one of the organizers of the NITO conference,
Yeah.
and just simply, again, a lot of the stupidity that happens on Twitter. Right? There was, when you were at the conference, you were live tweeting what was happening to you because you
Yeah.
were trying to ask. So it started with you trying to ask questions of the various speakers. Were you prohibited from asking questions, or did you get a question off first and then they took the microphone for you? From you?
No, what happened was that it was probably the second or third session before lunch on the first proper day. Zheng Lixi was the featured speaker, speaking remotely, as you said. We weren't explained why she was speaking remotely. But so she gave her presentation. It was fairly uninformative. And so I wanted to ask about the Mojiang miner. So the thing that was concerned, arguably of all the questions I wanted to ask her, I wanted to know what was why did they do such a superficial investigation of the disease contracted by these miners when it appeared to be that they had suffered from a novel coronavirus and potentially this was another SARS outbreak. So this is an outbreak in 2012, and the samples from the miners were sent to the Wuhan Institute of Virology. And our theory is that those samples are essentially what led to the SARS-2 outbreak, but there was a break because the Mojiang miner outbreak was in 2012. So we wanted to
Jonathan,
know. Yeah.
right. So that was going to be the transition to that Mojiang mine. Yeah. No, so that's a critical point about when we talk about your work on the Mojiang mine.
Yeah.
So you wanted to ask her about about whether there were other strains, or sorry, other samples
No,
that.
my question was essentially why they only took blood samples from those miners. What other samples did they take? If you want to know whether someone has a lung infection, a lung infecting virus, taking blood samples from those people is not very useful. And so to say that you can't find blood sample virus in their blood samples is probably not a very useful way to investigate that outbreak. But was that really all that was done? That seems ridiculous to us.
But you weren't, of course. So then you raised your hand at the end of her presentation,
Yeah.
and
So I never got to ask this question, right? All that happened was I raised my hand at the end of her presentation, along with a couple of other people in the audience, and someone with a student with a microphone came over to hand me the microphone, and they were just about to hand me the microphone, and all of a sudden they stopped, and it was clear that the people on the platform wanted me not to be given the microphone, but the microphone to be given to somebody at the back of the auditorium. So the student was quite confused, but they end up going to the back of the auditorium. And I found out later, because somebody was sitting closer to her, that Flo DeBar had told the student not to hand me the microphone. And obviously this was quite quiet, and so I didn't hear it. All I knew is that it was a rather odd sequence of events, that I was going to be get the microphone, and all of a sudden I didn't get it.
Right. So this begins a pattern of sort of, censorship or marginalization or deplatforming of you at that conference, correct?
Yeah, so when that person at the back of the auditorium, yeah, I mean, correct. And when that person at the back of the auditorium finished their question, I put up my hand straight away again, and another student came over to give me the microphone, and this time Flo DeBar very loudly says, "Don't give him the microphone." Right. And she's an audience member. She's not on the podium, but it's clear that the people on the podium didn't want me to get the microphone either. So they were trying to communicate with the students whose, standard job it is to go to hand out microphones, because they're young and athletic and they can lean over people and so forth. We always say, scientific meetings always have students handing out microphones, and they were being redirected away from me by either Flo DeBar or the podium.
Right. So I guess the it's incredible. This description to me was incredible at the time while I read it. But of course, when I first saw that, I did say, look, well, the organizers have every right, in my opinion, to not let somebody ask a question. But of course, that needs to be very clearly like that should be publicized, because they need to be held accountable for why they didn't let you ask a question. And to my knowledge, I'm not sure why. Was any reason given for why you weren't allowed to ask a question?
No.
Yeah. Okay. Sorry.
Nobody offered, and I didn't ask. I did ask why they didn't let me hand out papers, and they said the reason given was because I didn't apply to give a poster, and because I wasn't paid as I hadn't paid my way to be a commercial sponsor or anything of the meeting, which to me is totally bogus. I've handed out materials at dozens and dozens of meetings. There's nobody's ever given such a reason, and it's hard to imagine why that would be justified in the first place. Science is a place of free sharing of information, or it used to be. But it was clear too that the podium didn't want me to ask questions either. So at the podium, at the end of that session, before we were sort of halfway through the question period, and the person at the podium, who's a researcher at the CDC, said that they called David Wentworth, who I'd never heard of before. He said he was going to take the privilege of asking the next two questions, and then that would be the end of the questions for that session. And that's something I've never happened before. The people on the podium, they ask questions if no one in the audience asks a question, just to avoid embarrassing the speaker. That's really their role. It's not to prevent people in the audience from asking questions. That's unprecedented in my experience.
Well, it's not only unprecedented, but it's indefensible. And the fact that you live tweeted about it, and then also they stole your handouts, correct? And you had to chase them. Weren't they about to throw away your handouts, or they?
Well, so what happened was, I did, regardless of what they said, that whether I could share my handouts, I decided I was going to leave them out anyways. There were loads of empty tables at the meeting. It wasn't like I was displacing someone else's material. So I was leaving handouts where people, coming and going from the auditorium could see them. But I kept noticing that the the handout materials were always gone. So I'd leave out small piles, 5 or 10 or whatever of each paper, and then they would always be gone at the end of the session. So I go into I leave them out before the session. When I come back for the end of the session, they're not there anymore. So I thought, well, maybe conference attendees are taking them. But quite likely, I suspected that the organizers were taking them. So I thought I'll leave some out, and then I'll watch from a little distance to see who takes my papers. And it turned out it was the conference organizers who were taking my papers.
But then you chased them down the hall, right?
I wanted to. Well, I wanted them to know that I'd seen them, and I wanted to get them back again. And I wanted to see, firstly get them back, but also get the other ones that I'd taken back. And I wanted, because I thought I could, I thought they would throw them in the bin or something like that. But it turned out that they didn't. And so we had this sort of comical James Bond type thing where I chased them down the stairs, and they didn't realize they were being chased because, they just took or they just scooped up every single thing on the table, which was mine. And then they took it downstairs because that was the direction they were walking in. But they had no real reason. They weren't going anywhere. They were simply walking past my table, the table where I'd put the materials in order to remove my materials. But of course, when they gone another 50 yards down the stairs, they just turned back to go back to the thing and then didn't realize that I was there behind them trying to figure out what they were going to do with my materials, because I thought, well, I'll probably just throw them in the bin, or maybe they'll take them to the ladies' room and throw them in the bin, because they were both ladies. And so when they saw me, so they turned around because they weren't going anywhere. And so they turned around and almost bumped into me. And then they realized that they were carrying my materials, and that there I was and I'd seen them, and they turned around and fled in the other direction across the auditorium.
So I guess it sounded like you didn't want to get into the detail, but to me, this is the fact that it's so comedic, but it's unprecedented. It's embarrassing. I often use the word embarrassing. This is just simply embarrassing behavior for a scientific conference. And as I mentioned at the beginning, or 2 spaces ago, you're a virologist. Like you have a degree in virology. You have every right to be there. You are very knowledgeable about a lot of the subjects that are being presented there, especially COVID origins. And yet you weren't allowed to speak. You weren't allowed
to. Well, eventually, I did get to speak, but only by having my wits about me and so forth, and by being extremely persistent. We got to a stage at one of the sessions when I was the only person in the room with my hand up who wanted to ask a question. And they stopped the students from handing out microphones, because I guess it was too embarrassing to have them in real time telling the students not to let me have the microphone, and embarrassing to the students, to the audience. I mean, half the audience was quite perplexed about what was going on, and plenty of them heard Flo de Bar, and so so essentially they had conference organizers. Half the conference organizers that I could see ended up handing out microphones, subbing for the students, and the other half of the conference organizers were sweeping the halls trying to find my papers to clear up. So I was kind of a bit embattled at this point, when chasing these people down the hallway to see really for sure who was taking these documents. I needed the proof that they were taking the documents. But eventually I did get to speak, but only when I was the only person asking questions. So like I'd be the only person in a hall of 700, 800 people or whatever trying to ask a question, and they wouldn't come to me, because they've been told not to come to me. And people in the audience were pointing at me and saying, there's someone who wants to speak over there. And the people with the microphones would pretend not to understand them.
So well, first of all, I love that you're giving the oral history of this, because in my mind, I'm hoping maybe we can make like an animated visual of this, actually, like to have a visual to go along with the story. Before we move on to the Mojiang mine, I wanted to ask you, I guess I wanted to. What's that?
That was tick liquor.
That was tick liquor? You heard that?
Mm-hmm.
I don't think so. Can you hear me?
Yes.
Yes, we can hear you.
Oh, wow. Technology's amazing.
It's amazing. Yeah, this is an incredibly horrible format, but that's why I love it. So go ahead.
I just wanted to ask Jonathan if he was aware that that strain of virus that Fruehling and Baer used was the same strain or the same isolate that was used by Koopmans in her paper on European rabbit. And I think it's also used in an earlier study that Fruehling did with grad students. And I think Baer was, again, the anchor author on that, that looked at fruit bats and several other species as well. And I'm wondering whether that virus isolate was, it's BAV-PAT1. It's a very early German isolate that they may not have realized how the strains were working back when they started the experiment or when they did. But they certainly, by the time they published, they should have known that it was a D614G virus, but they never commented on that. But I'm not sure that they were completely aware of how that virus isolate, stood in the phylogeny of the of SARS-2 when they started the experiment. Just was curious if you had any comment on that.
I do. And the Marion Koopmans made the same comment on Twitter at one point. She said, this was, that was a strain we had available, basically. And that is true. That is what spread to Germany, and that is what was used in multiple experiments. But that doesn't excuse, firstly, not acknowledging that you weren't using an origin strain in the first place, and secondly, not repeating the experiment, because the Fruehling paper is extremely superficial. I mean, there's virtually no discussion in there about what their data means. A very, very small number of animals used, and so it's entirely a superficial experiment. You think of it as a pilot project, really, and there's no reason at all. I mean, even if you don't want to point the finger at Fruehling and their co-authors, there's ample reason to be repeating that experiment by them or by others.
Absolutely. Thank you.
So before we move on, I did want to ask one other question. Oh, thank you, Ticklicker. I'm going to remove you from the speakers. Thanks for your question. I wanted to ask you about why you didn't bring a friend to the meetings. How come you didn't come with a person that could sort of help you out get questions off? That was a question that actually Megan had.
Yeah, I did. I mean, I did need someone else. I'm not very tech savvy. It would have been great to have someone with a cell phone, great to have someone taking pictures, so on and so forth. But I'm just used to traveling to these meetings on my own, and I don't, I just, it's a very busy time. You're putting out papers, you're trying to listen to the talks, you're trying to take notes, you're trying to, then ended up trying to put it on Twitter. Absolutely could have done with another person, but there isn't, I mean, it costs a lot of money to go to these meetings. It's not a cheap trip. You have to stay somewhere. It costs thousands of dollars to do that. So it's not, it's not in our budget.
You did mention that there was one person that appreciated your questions that you met with at the meeting, right? You had a nice conversation with at least one person at that.
Yes, I did. Yeah. So this was a Spanish researcher who'd noted he was at NIDO, but it turned out he'd also been at the first meeting. So the way that I think about this is that there was a connection between these meetings in the sense that, I had asked this question of Professor Baer at the European Congress of Virology, and people like Volker Thiel did not want to repeat of that. because they knew that I could embarrass, a speaker in a scientific sense in a very severe way. I believe that a lot of people at the European Congress of Virology took home some important messages from my question. And I think that, if I'd been allowed to ask the right question of Zhengli Shi, the same thing would have happened. And it would have been very embarrassing for all of them, really, who are defending a zoonosis, that Zhengli Shi can't answer these questions, because there is no good answer to the question I was going to ask her about why you do such a superficial investigation of the virus that the miners are infected with.
So actually, when I hear you talk about this, and I had thought about this before, not making any promises, but I kind of really want to go to these virology conferences, not necessarily to, and I'll tell you why. Because one of the issues, of course, is identification of EPPP research, right? And it's right at this point, there's no way of anyone knowing who's carrying out EPPP research until it gets published. But at these conferences, it might be something where, by members of Biosafety Now going to them, it could be a way of sort of, engaging in that conversation, like injecting the concept of thinking about not only safety, but whether, because one of the things is like, why are some of these experiments even done? These are very dangerous. A lot of these are very dangerous experiments that I guess I would characterize as kind of stupid or useless, and they're really just done for a paper in a high-profile journal. But that's not. It's not an easy thing to communicate to the public. But as scientists, we very clearly see the fact that these experiments only get high-profile publications because of the way that the journal system works, and the sort of dangerous experiments somehow are viewed as extremely valuable, where they really have provided nothing in terms of developing therapeutics. So, but I would actually love to be at a conference with you. I think it would be fun, particularly with the question and answers, because you're very polite. I don't think I would be as polite with something like that. And maybe we could get a group of people to go to one of these next time. Not as again, not to be confrontational, but to actually, put people on the spot, to ask them the difficult questions, to demand that they answer them. Because this is ridiculous. What you describe is embarrassing, ridiculous. It's, we're beyond saying it's not scientific. I mean, it's something very nefarious. It's intentional censorship of somebody that with very basic questions about the origin of COVID at a conference where we then later learned the FBI was interviewing some of the scientists about the origin of the virus as well. So it's absurd. So anyway, I guess maybe you wouldn't even want to be around me. Maybe you would ostracize and censor
I do in principle. I make it an important principle to be polite in these questions.
I know.
I really put a lot of effort into that.
No, I mean, actually, it's very commendable. I find it, I mean, that's something that I would find extremely difficult, and it really needs people like you to put yourself out there where, you're getting mocked. But you're just very, you're calm, you maintain your composure, and you ask your question. Like what you did at the Pathogens Project, and that people can see that online, where you asked, you spent 2 minutes making your statement, and they tried to cut you off, and you said, no, this I'm going to finish this, and you did. I think that's why I was very happy to have you on one of these space and to actually get to talk to you about this, because voices like yours are very important, and they're being completely, not even just marginalized, just like completely silenced in this space, which is absurd. So anyway, I'll get off my soapbox, and I want to now go to your work on the Mojiang mine. So I was interested in this because I learned a lot about this over the last 2 days, because when I spoke with you, I didn't appreciate that you actually have, I guess one might consider, a pretty parsimonious, or the most parsimonious explanation for how we got SARS-CoV-2. Is that a fair characterization of your proposal?
I think so.
Yeah, and so could you explain to people what your explanation for how we got SARS-CoV-2 is? and why it's so parsimonious?
Yeah, so the parsimony is a little more complicated, but the simple explanation that we offer is that in 2012, 6 miners became sick who were shoveling bat guano in a cave in Mojiang County in southern Yunnan, and they all became sick. 3 of them died, but the 6 of them were all taken to a hospital in Kunming University Hospital, and they were kept alive on life support with seemed to have been immunocompromised by their experience. And the Wuhan Institute of Virology took blood samples and other samples, probably, but we know blood samples from those miners and took them back to Wuhan. And our theory is that a bat coronavirus from that locality. And we know, for example, that the nearest, rat G13, which for the longest time was the nearest identical wild virus to SARS-2, was also isolated subsequently in that mine. But our argument is that the bat virus became adapted to humans and to human lungs inside the lungs of those miners, and those miners suffered, we know, from very similar, very COVID-like symptoms, and that the Wuhan Institute of Virology essentially was sampling a human-adapted, lung-adapted virus, and what they ultimately sampled was in fact SARS-CoV-2.
So there's a couple things to sort of try, I guess, for people that aren't for people that aren't up to speed sort of on the biology and the idea of, what can happen inside them. Because part of this, it involves what happens inside a person that's infected with the virus when they have, a long-term infection that isn't cleared, or they have a compromised immune system, right? So that's essential to this proposal. I think actually I would kind of like to go into this in more detail because it relates to, discussions about how the original Omicron variant emerged. And I think it's, conceptually, it's a bit difficult to understand what people are saying in terms of the mechanism for how you see emergence of different variants in a person.
So the comparison point, the reference point is that in the general population, people are infected with a virus. They catch more or less a uniform variant of the virus. They get maybe 10 identical SARS-2 viruses. And after 10 days or so, they transmit them, or that line goes extinct. And what they transmit, for the most part, is an identical virus. So evolution under those circumstances is quite slow. If you imagine that the virus infects an immunocompromised person, so maybe they have HIV, maybe they're on cancer treatment, or maybe
can you back up for 1 second with this? What you said? You said evolution under those circumstances is slow.
Yeah.
You mean that you don't get evolution under those circumstances, or that there's actually
you do. I mean, that is the background rate of the evolution of the virus. But that background rate is something like 1 mutation every 2 weeks across each lineage.
Right. Sorry. Go ahead. To me, this is actually quite complicated. And it's extremely important. That's why I was trying to.
Well, it's important to the theory, but it's also important to the origins of all these novel variants too. Yeah. So the idea is that when a virus infects a person who's immunocompromised, that virus can be in that immunocompromised person for many months. And what that means is that instead of the virus passing through a bottleneck every 10 days or so when it has to transmit to a new person, so you get 10 copies of the virus, you transmit 10 copies of the virus, often they're all identical and often either identical to each other or identical to the one that you got. Right? In an immunocompromised person, what happens is the virus infects that person, and then their immune system, however weakly, kicks in, but it fails to clear the virus. So the virus will mutate inside that person, It can potentially recombine inside that person. It can potentially spread to different body compartments, so you have different forms of the virus in different parts of the body. And it can also be involved in a kind of struggle with the immune system, because the immune system learns to recognize after a while the virus that the patient has. And so there's sets up a selection pressure, and that selection pressure is normally absent in a normal virus infection. If you transmit the virus, say after a week or 5 days, your immune system hasn't even kicked in. Right at that point, your acquired immune system hasn't kicked in, so there's no selection pressure for the for variants that resist immunity. And so what's happening in the circumstance of an immunocompromised person is actually that the virus, because of recombination between viruses, because of the mutations that can build up, and because of the number of those mutations that can build up, the virus actually evolves much faster inside an immunocompromised patient than the entire rest of the pandemic put together. And this is the origin, it seems, of the alpha variant, the beta variant, the Omicron variant, and many of the other variants that have taken everyone by surprise by, firstly, the number of mutations that they contain, but also their ability to evade immunity.
So just to clarify, this is something you said it was surprising because is this something that has not been observed previously with other viruses? This type of
that's broadly correct. Yes,
okay.
yes. I mean, it was the origin of these variants was took scientists by surprise, basically. Yeah.
Right. So I guess that gets into like the phylogenetics and looking at the sequences and noticing that it looks like it's coming, that you see these crazy variants emerge that
Mm-hmm.
look like they're derived from something that has been gone for 4 to 6 months, right?
Yeah.
That's kind of the idea, where
Yeah.
all of a sudden something pops up, and it only makes sense if you believe it emerged from a progenitor that's been hiding for a while. And so
Yeah.
then, but they've
Yeah.
been able to then in a lab, right? They can mimic this in a lab to show
Well,
that this will
to me, the
work.
most important experiments are where people have, initially by accident, registered the fact that the same immunocompromised patient keeps coming back to the hospital, and they keep being sequenced, and they have a different version of the virus every time, but it's clear that in each case the ancestor was the one they had before, so they have basically kept the same virus for months and months and months inside them, and it has evolved. So that's the most compelling part of that story, for my mind. So the fact that these mutations have been mimicked in labs is not of such of interest to me.
Well, okay, so going back then to the mine, because so to go through some of some of the ramifications of your model. One, I guess, as an aside, is that you are comfortable saying that Omicron, the original Omicron, emerged through this process, because some have suggested that instead it was derived from intentional manipulation in a lab. Um, so that's one aspect of this, and I'll sort of push that off to the side. The other key point is that your proposal does not require there to be an intermediate animal host. That
That's
your
correct. Yeah.
proposal is a direct line from bats to humans, which involves this extensive recombination happening in one of the miners who is, you propose, with a compromised immune system. So the idea is that, maybe correct me if I'm wrong, that a miner gets sick, and then that miner is, now, do we actually know that the miners themselves were, in a long-term state or of immunosuppression with infection, or maybe actually.
I would say we do. We do know that because they acquired a lot of infections in the process. They were continually being tested for the bacteria they had and the fungi they had and so forth. And many of them developed diseases that are characteristic of people who are immunocompromised.
Okay, so this proposal also involves that there is multiple, or sort of, you have SARS-CoV-2 emerging as I guess, I don't know if you use the term, or a mosaic is what you use, a mosaic that represents the combination of several different progenitors, correct?
Yeah, we don't know what initial viruses the miners were exposed to, right? So what we do know is that phylogenetically, SARS-2 has many parents. There are viruses collected in the general vicinity of the mine in northern Laos, within a few hundred miles of the mine, that are all, to some degree, the grandparents of SARS-2. They are the most closely related across certain parts of the genome. For some it's the spike, for some it's the N protein, for some it's small regions in other parts of the genome. So you've got a whole set of wild viruses that are the progenitors of the virus, and what you have is essentially evidence of widespread recombination. But the question in part is, did that recombination happen before the virus entered the miners, or did it happen inside the miners? And we can't answer that question, except that we do know that it happened.
I want to perhaps come back to this point because obviously even though you're right, I said it was parsimonious, but at the same time, there's a lot of complexity. I think the critical thing to me in terms of being parsimonious is you are proposing a direct bat to human transfer, which then and then the way it gets to Wuhan is through a collection of a sample from an infected miner, transport to the Wuhan Institute of Virology, where through some lab manipulation it gets out. Is that
Yeah.
simple?
Yeah,
Okay.
yeah. So the theory is simple, but it's
Simple, yeah.
parsimonious in the sense that it explains the phylogeographic origins in southern Yunnan, explains emergence in Wuhan, it explains the lung preferences of the virus, it explains the human adaptiveness, and so on and so forth.
Well, but there's one key thing that it may not explain. And it's the furin cleavage site. Now, so you have an answer to that. What is your explanation
Yeah,
for
we do.
the
I mean, it is.
unusual codon usage of the furin cleavage site in particular?
It
So
would be. It would be a what? Sorry.
I'm sorry. What I meant was, so the one thing, so all this is well and good until I think that the furin cleavage site is a bit of a challenge for this model. So you said that you have an explanation. So what is the explanation?
So what we think happened is that there is a human epithelial protein called ENaC alpha, which has 8 amino acid identity at a region that largely overlaps the what's called the furin cleavage site insertion of SARS-2, the 12 nucleotides that it acquired compared to the nearest ancestors, who are rat G13 and banal 52. So what we think happened is that there was a recombination event between the progenitor and this ENaC alpha, which generated a furin cleavage site. So the sequence in ENaC alpha is a furin cleavage site itself. It recombines with SARS-2 to generate a furin cleavage site, but it generates a furin cleavage site with an extra amino acid. So if you look at what's called the furin cleavage site, the insertion is a PRRA, and only the RRA is actually necessary for furin cleavage. So what we are proposing is that the recombination with ENaC generated the furin cleavage site, and then the P of PRRA actually mutated afterwards. So it was a two-step. What we propose is a two-step model. There's recombination with ENaC alpha, and then there is the acquisition of the P, which is simply a substitution stage. The proline is more or less conserved through the whole history of the pandemic. So that means that it's not being lost, right? And if various lab experiments have been done to mutate the proline, and what mutating the proline does is it limits access through the early pathway, and the early pathway is the lung pathway that the virus is basically switching to. So the ancestors, the progenitors, all use the late pathway to get into cells, but the late pathway is not very active in lungs, and so you need a virus. Basically, there's a selection pressure for the virus to acquire a means of entering the early pathway. And our theory says that the furin cleavage site is necessary to enter the early pathway, but it was not sufficient for it to really work well. And so the proline is a second step that was selected to improve the access to the early pathway and to make it really a viable pandemic virus.
So you explain the proline, and then you also have explained the unusual codon usage, because that's now simply coming from the codons that are in humans, correct? From a protein.
Yeah, there's a little bit of a mismatch between the codon usage. So there is you can ask questions about that, I think, and I don't think that we don't really have anything to say about the usual codon usage, except that it's something that would have been presumably is adaptive, but we don't know.
So so that might be more of a concern with regard to this then. So certainly the proline makes sense, but why you would have codon changes for the and I guess for people, you'll have to remind me, what are the codons that are in the virus, and then what are the codons? These are the two codons.
I don't have that information in front of me, so I'm not in my office.
Justin, do you remember this? Is it CGG? Is that what it is? Some people know this off the top of their head. I'm in the parking lot, so I can't go to my computer. I have to. Do you remember, Justin? Oh no, we don't know this. Okay, let's just.
No,
I think it's CGD.
It's something like that.
Okay.
But we don't ascribe any great significance to that in our theory. We don't think it's really material.
Okay. Well, so basically, yeah, I just wanted to I like this, just trying to step through this, because in a way I said it's parsimonious, and it does explain the lack of an intermediate animal. It does make it a little bit easier to understand how, how you got the viruses, the closest progenitors are all within the region of this cave. And how those, we know there was sampling happening there, and we know that those were brought back to the Wuhan Institute of Virology. So you have a lot of the pieces that could explain this. And interestingly, you don't have to invoke any gain of function experiments. You don't even need the diffuse proposal, correct, for this model. You don't.
That is correct. It's not necessary, but there's room for some engineering possibilities, like for example, modifying silent codons for reverse genetics and so forth. So we don't exclude that possibility. No.
But it would be of use, though, again, to have this discussed, publicly, so people can sort of, poke the tires a little bit. But so the question then is, so what have your critics, or, I know that you posted this article.
Can I ask a question about that first? So, the ENaC alpha, it has a shared amino acid sequence, but is that sequence of the human ENaC alpha gene, like the nucleotide, like the DNA nucleotide sequence, the same as in SARS-CoV-2?
Over some of that stretch. So there's 8 amino acids that are identical, and then for about half of that whole sequence, the nucleotides are identical. Yeah.
Okay, and the other question, I've actually always been skeptical of explanations for the furin cleavage site through recombination, just because it seems like such a short sequence, right? So if you get recombination, it matches the left half of a genome with the right half of a genome, right?
Mm-hmm.
And you can get segments, but you need 2 recombination events, and usually they're not that close together.
Mm-hmm.
It's more like hundreds of nucleotides, right? Or
KB. There is a paper from Thomas Peacock, which is it's in Virological. Actually, it's not strictly a paper, but they go through all the evidence for different that some of the changes that have emerged in sequence variants over the pandemic, and not necessarily variants of concern, but all the different genome sequences that have been generated, the millions of genome sequences, and how many of them have evidence of recombination with human messenger RNAs or other kinds of RNAs. And there's a reasonable number of them. There's about half a dozen or so. And they're very short. They're all very short. They're all sort of 10 to 20 nucleotides. So it definitely can happen.
Okay. So I mean, another possibility is just de novo insertions, and I know that de novo insertions of similar length can happen. Anyway, that's always what I thought was more plausible. That there's that the furin cleavage site. So even if it was genetically engineered into SARS-CoV-2, it had to come from somewhere. And well, I mean, there are other like coronaviruses that have that have been observed to have de novo inserted furin cleavage sites.
Mm-hmm.
But I think it's there's arguments on both sides. I did want to ask you though, Jonathan, like, so would the diffuse proposal play any role in your model?
Not specifically. I mean, it's a while since I read it, but the difficult. Initially I was attracted to that because there's a discussion in there about adding human-specific furin cleavage sites, right? And the RRAR is a human-specific cleavage site because it's present in ENaC alpha. So it would be a model, as Harrison and Sachs suggested. There's a clear precedent for using that furin cleavage, that specific furin cleavage sequence. But then the issue is twofold for us about why you would. There are actually four arguments why we think it was not engineered. But the first one is that normally when people have engineered furin cleavage sites into coronaviruses and other viruses too, normally you don't do an insertion, you make substitutions. And the reason for making substitutions is that they're basically more conservative. You're not going to disrupt the function of the whole protein by adding 4 amino acids, which you might well do, right? So, a researcher wants to do something conservative, and that's historically what's been done. The only time that people have made insertions, except with one exception, the only time people have made actual insertions as opposed to substitutions is when they were trying to model a furin cleavage site that had already arisen naturally by presumably some recombination event. So that's one reason. It's not the scientifically logical way to add a furin cleavage site if you want to get your paper published. And then the second reason is that why would you add the P? Like, what's the P doesn't play a role in furin cleavage per se. So why would the Ralph Barricks or Zhengli Shis of this world add a P? There is no logical reason why they would want to do that we know of. And then the third reason is that this site has been conserved so precisely over the whole history of the pandemic. I mean, there are one or two variants which have changed the P into an H or an R, and then the other variants arrive where the P is back again. And so this, even the P is really, really, really highly conserved inside these viruses. And the question is some, genetic engineering mastermind really clever enough to organize the perfect furin cleavage
So, Jonathan,
site?
I've got to disagree with you. That P is mutated in all the major lineages, like either to an H. Well, at least that's what I'm getting on the covariance site. Like that P is
The alpha changed it into an H, I think, but then the alpha was superseded by another variant that where the P was back again. So yes, it's not a no-brainer. But the other complication to all this is some of these mutations are dependent on their context, and the P may be useful in alpha, or the P may be less useful in alpha, and then become more useful to another variant that emerges later. So there's a little wiggle room in this argument that is hard to, I don't think it's I agree with you. It's not a definitive argument, but it's incredible degree of conservation, and it's seeming unnecessarity for the furin cleavage site itself means that a genetic engineer is very unlikely to add it in the first place.
Well, the one thing I want to just point out, though, like it's, so in the diffuse proposal, they talk about trying just a lot of different things, right?
Yeah.
And that's usually what genetic engineers do. You can't predict what a specific sequence is going to do, so you just try a bunch of things. In SARS, and in the diffuse proposal, what they were proposing to do is doing deep sequencing of spike proteins present in samples, and in finding low abundance but presumably high risk variants, and inserting those, and then putting those variants into the spike protein, the potential cleavage sites, into spike proteins of a low risk but high prevalence variant in their samples. And I don't know. So that's always been, that's always that's my current model of it. I'm just trying to bounce this back and forth with you.
Pause you to insert one thing, which is that the P is not a substitution, right? The P in their description, most of this deep sequencing is about alternative bases and alternative amino acids. And most of it doesn't involve substitution. Substitutions are a different, insertions are a different order. Yeah, pardon me. Insertions are a different order of change than as a substitution. Because you got to add 3 nucleotides, right, as opposed to change one into another one. So that you got to ask a slightly different question.
Well, I think from the diffuse proposal, the way I read it is that what they plan to do is like sequence a lot of spike proteins, like the spike proteins in bulk, deeply in their samples. And if like one of those spike proteins has very low abundance, does have the 4 amino acid insertion with the P, and the PARR, the PRRA, then that might be where they got it from. But I think it's good to have these discussions and to throw these ideas back and forth. It really frustrates me that there's no discussion of this in the virology community about like what is the most like if this was a lab leak, what might have happened, because there's so much information about there, but the community just seems to treat it like a conspiracy theory and not like an actually important question to answer about where this pandemic came from.
There is one fairly recent discussion. It's kind of a scientific op-ed that was published in multiple journals at the same time, which kind of went through some of these points. Why would they add a P? Why would they make an insertion? So belatedly, that discussion sort of had. Pardon me. I forget the name of the authors. Yeah, I think it is, and I think it's published in the 3 or 4 journals, and each one is the senior editor of that journal. So they're basically plagiarizing themselves.
Yeah, I have a lot of issues with that.
Of course. But at least they're having that conversation, sort of.
Well, it sounds to me like in that paper they're just repeating proximal origins mostly, and just coming up with the same arguments. that the Warby et al. And Pecker
Yeah.
et al. And Anderson et al. groups
Mm.
do. But I don't
Yeah.
want to get too
It's
much
not a
into
good
the
paper.
weeds.
Yeah.
Thanks for this conversation. But here, Bryce, why don't I turn this back to you?
You're getting in the weeds, Justin. You're getting in
Yeah,
the.
I know. I
No,
like
no, you're fine. Well, no. That's the thing. Like he can't go to the scientific conferences and get in the weeds, so we got to do it on an X space. That's what we're for. We're here for. So I guess. So bottom line, I guess this is the thing. Like to try to get out, look at a like more of a forest level view. You have a proposal for how SARS-CoV-2 emerged that actually isn't all that super conspiratorial and doesn't involve a lot of nefarious actions. It really simply involves people isolating viruses from miners that got sick and some of them died, bringing them back to the lab to study, and then it gets out. So it's actually, in a way, it's less, there's a lot less going on there. I did want to go back. Is that, I guess I would say, is that a fair assessment of your, like a very.
To a point, I mean, it does involve the Wuhan Institute of Virology being extremely disingenuous.
Of course, yeah, that's all baked in. But I'm just saying that there's less going on. I did want to go back real quick to the fact, like, your role in the actual discovery of that mine in the first place, because I completely forgot to bring that up at the beginning of this discussion. So you had said, can you explain how you first got interested in the Mojiang mine?
We were sent a copy of the thesis, I think by Manali Rahalkar. So she already so the seeker found the thesis, the master's thesis, which was basically the doctor who treated the miners. So this was a catalytic discovery for us.
So this
So
is
the
just to
seeker
clarify for
found
people.
this,
Yeah,
downloaded the speaker. Yeah.
the people that don't know. So the seeker is a member of Drastic, who is sort of anonymous, sort of isn't, right? I mean, but and he and also Mona Lee is also a member of Drastic, and they. So sorry, I just wanted to fill in for people because the seeker was gone from Twitter for a while. Actually,
Yeah,
appeared
yeah,
recently.
a
Sorry.
long time. So the seeker found the thesis and translated bits of it using Google Translate and so forth. But we wanted to make a bona fide proper copy of the thesis with the whole thing there. So we downloaded our own copy of it, and we got a professional translator to make a translation of it. And so we wrote it up as with all the T's crossed and so forth, because we wanted to make sure that there's not any information missing from this thesis. So that took us a little bit of a while, but immediately reading the thesis, it occurred to us that these miners had COVID-19. It really looks so much like they had COVID-19. And when you read at the end of the thesis that the expectation of the doctor was that they had a novel coronavirus, then, to me, it was very obvious what ought to be proposed next.
So I guess, yeah, and we're really just only touching the surface of a lot of this because, as you pointed out, the disingenuous behavior and the lack of transparency is all baked into this. And I guess we're speculating at this point, you're saying all the way back to the first discovery of the miners, because that wasn't, when was it, I guess, when did the public become aware of those miners? This is
I
in,
think it was. It was the seeker, if not drastic,
and that was in
because
2020 or late
2020. Yes. So we wrote up our proposition in July, and we became aware of it in late April or early May, something like that. And so we, but the Wuhan Institute of Virology never acknowledged that the rat G13, the nearest virus that they found published at the same time as SARS-2 sequence itself was obtained in a mine where because they was looking at the origins of a mine outbreak. They never said that.
Right, and that was collected later on. Not it was collected or at
It
the
was
same time.
collected 6 months later,
6
roughly.
months later. Right. Okay. I want to go. So I'm going to have Jason. You want to ask a question? I'm going to add you as a speaker. You have a question for Jonathan, if you're able to. Okay, Jason, can you hear me?
Was that a question?
Nope, that was a troll. That was good, I guess.
Interesting.
You attracted the trollers. That I didn't understand. That was good. I don't know. We did get a troll last time too. I don't know. It doesn't. I don't know. Anyway, apologies. That's part of the thing about welcoming everybody to these spaces. You do get. All right. So let's see. We're gonna try Heath. Now, Heath, if you're gonna troll, I at least want to understand what you're saying, because I did not understand what the last person was saying. All right. Here we go. We're gonna see if Heath Fuqua is gonna ask a legit question. Here we go. Heath.
So my question is,
Oh, thank God. You're a legit person, right?
yeah,
Okay,
legit
good.
person.
All right.
How can we distinguish between serial passaging and this natural spillover hypothesis?
I would answer that in several different ways. If you have serial passaging, you need serial passaging that has to meet several criteria. So one is that the virus that emerged was very well able to combat the acquired immune system. Right. So if you serially passage a virus, the normal series of events that happens is the virus becomes adapted to the cell line, for example, that you are using. So the cell line, so the virus loses its ability, for example, to combat the immune system, because the virus has, various clever strategies by which it, sneaks around cells and different routes by which it gets out and so forth. The biological requirements for avoiding the immune system are very acute. And so what you need to imagine is that it will be very difficult to create such a virus simply in cell culture. So then the question becomes, can you create a exquisitely well human-adapted virus by passaging in a whole species, right? And so you've got the problem that the ACE2 receptors are always different. You've got, would it be a macaque? Would it be a rabbit? Would it be a humanized mouse? All of these serial passaging opportunities are difficult to envisage that end up in the place of a very well adapted of a virus that's very well adapted to humans. So the ease with which SARS-2 broke out in Wuhan and spread, all around the world is testimony to it being road tested in a human. So that is the basis of the argument.
Can I follow up there?
For sure.
So how about a chimeric virus that is passaged in, say, ACE2 humanized mice? Wouldn't that possibly explain the ability of the virus to kind of hit the ground running and not have to ramp up so much if it's already been sort of exposed to the same, really attuned to these receptor sites?
I mean, you can imagine, the humanized ACE2 part of the mouse solves the problem of the adaptation, presumably does anyway, solves the problem of adaptation to the ACE2 receptor. But then you've got all the other the virus has adaptations to humans, presumably spread all through it. And so you have that. You basically want, I'm sorry, I'm a little tired. So what I think is, the experiment that a person would want to do, I think, to rule out that possibility, it would be to passage SARS-2, the original strain, in mice in a humanized mice and see where the mutations arise, right? Because if mutations arise that are adaptive, that would imply that the virus was not passaged in that species. That would be the experiment to do the test. So the being passaged in a real animal does answer some of the questions, but not all of them, in my opinion.
Is that
It's a good question.
all right? Thank you, Heath, and thank you for not trolling us. That's great. Does anyone else have any questions? I guess if not, I'll well, I will say if Sam Husseini would like to ask a question, as he knows he is banned from asking questions, so he cannot. But anyone other than Sam can. I'm just teasing. I'd love for Sam to ask a question, but we gotta have now. Gregor, easy on the Ebola conversation because I know you're gonna want to do. All right, go ahead. Yes.
Yeah, since we
Gregor,
have both Sam and Jonathan in the room, I want to thank them for the super long article on Ebola. It proved to be a very useful backbone for my own work. So thanks to both of you.
Gregor, you just wanted to thank them.
Yeah, I wanted to thank them. It was a really important article. I found it very useful for building my own work on it.
Okay, well, thank you. Well, thank you for thanking them. And we got to keep getting the proximal origin petition going. It's been a little bit stale. You got to keep sharing that as much as you can.
Yeah, I could share it again. You're right.
Well, no, it's in a phase that we're going to bring that back a little bit later. Don't worry. Like I've said before, that's not something we did for performance art. I will make that happen if it's the last. Well, either I'm going to be trying to make that happen up until I get thrown into the van and disappear.
Oh, another thing. So, if you want to make a space about Ebola, also consider hitting me up for some inside info. I made quite a lot of progress there.
Okay. Well, thanks, Gregor. I don't know. Sam, did you want to say anything?
Just to thank you for the discussion. And I particularly found, Justin querying Jonathan, very educational for me, since that's not my area of expertise about the Mojiang miners and how efficacious it is and so on. So to the point about asking questions and so on, I felt obviously a kinship with Jonathan and his asking questions at scientific conferences and what I've tried to do at the State Department and elsewhere. And I think it's a different dynamic. When I'm at the State Department, you got to get your point across, and it's a much more confrontational thing than scientific conferences, I imagine. I would encourage, if anybody's doing this, if you don't have somebody doing camera work for you, or the organizers aren't having camera work, that you could, and if you have a phone or a device, you can just do the audio at least, which is useful in and of itself. So I would recommend that for any future work on that. But I mean, to my mind, asking tough questions in such venues, the more, at least constructively confrontational that room is, the more peaceful the outside world is. That is, if it's schmoozing and snoozing at a, for example, State Department news conference, you're probably guaranteeing worse things going on in the outside world. So that's what motivates me in terms of part of what motivates me in terms of trying to ask tough questions in a context like that.
Well, Sam, what's amazing is that the representative for the State Department still calls on you.
Yeah, but the
virologists won't call on Jonathan. They won't even call on him.
But in part because we're operating under different parameters. That is, their job is to call on me to say that they've called on me, right? Because if they don't, then I go up to them afterwards and I embarrass them in front of other reporters and say you didn't call on me, which I've had to do. But they want to call on me and then cut it off as quickly as possible. And so it's a different dynamic. I think if they actually gave Jonathan the mic at a scientific forum, that there would at least they wouldn't be able to try to cut it off in 20 seconds,
Right.
So
Well, he's not even asking anything that, I mean, what's crazy is he's asking sort of basic questions
agreed.
that should be asked at scientific. I mean, you're asking questions about like that touch on like national security issues, and
Right.
he wants to ask questions about what BSL level they did an experiment in. You know, I mean, it's
Agreed.
ridiculous.
Agreed.
Yeah.
Although my last question was just simply, did USAID fund the Wuhan Institute of Virology, basically?
No, right.
You know, so yeah.
Which wait, now what happened? Okay, so for people that don't know, Sam had this very interesting interaction with, what is his name, Mike?
Matthew Miller is the latest guy.
Matthew Miller, very nice hair, I gotta say. And he, you know, you asked this question, and while you were asking it, he let out this extremely loud and rude sigh, which was
Right.
incredible.
Yeah, that was part of it.
And then can you explain what the question was that you asked him? And it happened right after the CIA report came out. Now I'm actually jumbled in my mind. What was the?
Well, yeah, the CIA report came out, but you know, there had been some other movement as well, and also critically, the State Department.
The Deep Vision had been canceled, right?
Correct.
And then that's right. So then there was the Weistrip committee released that CIA thing, which
Correct.
you suspect was done to distract from the Deep Vision being canceled, right?
I'm not sure. I'm not.
Oh,
it's hard for me to say about that. That it's, you know, think something I might think, but
I'm trying to out Sam you. You know, like
yeah,
I'm trying
right.
to like outflank you.
Yeah, no, I don't know. I haven't looked at the CIA stuff sufficiently, to be honest. But they, you know, the Lancet put out a report that they canceled their longstanding, you know, virus collection activity very quietly. And so I wanted to highlight that they'd done that and to get them, you know, I mean, this is the fourth question I've asked about USAID's funding of, you know, relating to the origins of COVID. And for the fourth time, they have ducked any substantial response. So that's where it stands right now.
Right, and they didn't even. So they answered you by saying you were being disrespectful or something, right?
In the end, you know, he was like, I disagree with the premise of your question,
right?
And then when I tried to follow up, they said you're being disrespectful, and
Well, I think you did. So okay, for those, if people want to go and watch the tape, I think,
yeah,
and actually the person formerly known as Fuzi who just arrived, I think I agree with what formerly Fuzi said, which is that you threw in the bioweapon thing, which then allowed them to not answer it.
Yeah, right? Arguably, but you know, you know, there's so many constraints. That is, if I just say, which I might, but if I just say, did you fund the Wuhan Institute of Virology? Realistically, it's going to just simply be weaponized to bash China some more. You know, so saying Ralph Barrick and saying, you know,
right.
You know, I mean, maybe people would argue that I should have just said gain of function work rather than say weaponized. But you know, you're
well, no, I mean,
you know, other people are welcome to go to the State Department.
No, I mean, I think that was why he was able to deflect it. That's all I meant.
Possibly.
Yeah.
Possibly.
Now, Fuzi, did you have, or COVID, or whatever that you're calling yourself now, but formerly Fuzi, do you have a question?
Yeah, well, I have maybe a quick comment. One is I just want to thank Jonathan and Sam for the Ebola piece. That's I think it's probably new for a lot of people. It's a good, very good exposure to the potential lab leak in Africa in 2014 that probably most people don't know about, including myself. Back to the Mojiang mine, my comment is that given the diffuse proposal, as a non-biologist, I'm still leaning towards it's genetically manipulated, having the FCS in SARS-2, given it is the only beta coronavirus have this feature. I understand Jonathan's point that it could evolve in humans and whatnot, but just giving the 4 minors, then you have a unique feature right away, instead of having infect the millions and come up with a unique feature. So just based on that alone, I'm leaning towards genetic manipulation in Wuhan labs for FCSC to emerge. Also, the U.S. government cables, they stated the Wuhan lab collected multiple SARS virus that can infect human lung ACE2 receptor directly. So they have a good batch of a virus that was ready to go to infect humans. They just make it more infectious with add-on genetic features. So that's my comment. I don't know if either Jonathan or Justin have any feedbacks on that. Thank you.
I have a comment, which is that if you accept that Omicron and Alpha and Beta and Mu and all these other variants arose in individual patients, right, which seems overwhelmingly the most likely explanation, then what that means is that the evolutionary rate of a coronavirus inside an individual person ends up being twice the speed, at least, of the evolutionary rate of the virus in billions of other people. So that is the observation that follows from those variants. And so that is I wonder if you have a misconception about the possibilities of what goes on in an individual person, because it sounds utterly counterintuitive that a virus in an individual person could evolve quicker than the rest of the pandemic put together. But that is the implication of these variants, because what happens in an individual patient is there are opportunities. So mutations arise, therefore diversity arises, therefore there's an opportunity for recombination between all these diverse types. So you've got this kind of like trial and error system that builds up inside these patients that allows the virus to evolve much more quickly than it does in even billions of other individuals. So that's our thinking about this.
Jonathan, I do have a counterpoint, which is that this cannot be the first time people go gather bear canoe at a bear cave somewhere. It's probably multiple times has happened before, right?
Mm-hmm.
So if FCS can arise so easily out of 1 out of 4 minors, then it probably has arisen out of those previous bear minors before. So how come this is the first time we see FCS in COVID, in a pandemic, SARS virus pandemic? Then you would imagine pandemic should have occurred multiple times with FCS before this time. I'm just saying we are playing numbers game. If it's as easily as you said it is, then you should have actually seen it earlier, maybe in SARS-1 pandemic. But I don't know. I'm just saying, I'm saying this out loud as sort of logical deduction. I don't have no favorite theory either way. Yeah.
I mean, the way that we would be thinking of it is that, the really important point is that these miners were taken into the hospital and kept essentially on life support. I think that you're probably right. A lot of people are getting infected by corona type virus, similar bat coronaviruses out in the wild. The prevalence of people who have some kind of immunity to these viruses suggests that individual infections are relatively common in rural populations. But what matters with these miners is, firstly, they get a huge dose. They're forced to work in this mine. They're there for 14 days. They get sicker and sicker and finally go to the hospital. But then when they get to the hospital, they are then kept on basically on life support. Right, so the hospital incubation period plays a crucial role in the theory. What presumably happens to most people when they come in contact with bat viruses, either they get a fairly superficial infection, or there's somebody, like you say, who harvests bat guano, but for some reason their dose is a little bit lower or something like that. So I know they're not in an enclosed space, or well, that wouldn't probably apply to most guano harvesting. But also maybe they get to finish when they feel sick. They get to go home because they're not essentially in a forced labor situation. So that's not a full answer to your question, but it's maybe a part.
So, so if I can just comment on that,
if I can jump back in on another point, Bryce, if that makes sense.
Sorry, that's Justin. But yeah, go ahead, Sam. Justin, can you pause for 1 second?
Yeah, go ahead, Sam.
No. If he wants to continue, I was going to raise another issue.
Oh, I was just going to say that I think former Fuzi's
should I just continue?
Point is perfectly fair.
When Jonathan was talking earlier about the conferences that he was going to and so on, mentioning a bunch of people, I jumped into this fairly early, and part of what struck me is that a lot of people who knew better, like Mark Lipsitch, for example, were incredibly silent for like a year, if not more. And that was very destructive for this process. I feel like they need to explain that. If the global public had been made to understand that lab origin was a very serious possibility in early 2020, a lot of people weren't aware of the possibility, and that's perfectly fine. But some people were well aware of the possibility and chose to stay silent. Obviously, Ebright and Francis Boyle and others were out there and getting abuse for it. But it was very concerning to me when a lot of people who had been warning exactly about this kind of thing happening were silent when it most mattered, and now they want to sort of come back in and dominate the narrative. And I think that's very problematic. And I don't know if other people have a view on that, or similar experiences or different experiences.
Well, so Sam, I think Sam. So I think I kind of agree with you. Like, I think there's a lot of people. What happened really early in the pandemic with the scientific narrative was very destructive. There was a small group of people who decided to very aggressively push the natural origins narrative, and a far, far larger group of eminent scientists knew that a laboratory origin was a possibility, and they chose to keep silent. And I think we're still recovering from that. I think, and I think it would. I don't know. So, Sam, I think it would make a lot of sense for you to directly ask those people, why did they keep silent when they were warning about this for years? And, I, for one, would really like to hear, I think a lot of good would be done if just more eminent scientists would speak out about this, saying this really could have come from a lab. And and that possibility is something we really need to be talking about, and not just treating it as an impolite topic that we can't bring up in conferences.
If I could chime in there on that point, I think it's a reasonable hypothesis that in addition to some of the grant funding capture that has taken place pretty obviously, given the surfaced emails, that there's also, considering the dual use nature of gain of function research, there's a national security implication here. And so you kind of get the double whammy on some of these investigators who privately express different opinions than they're willing to publicly express, because they recognize that sort of Damocles is hanging over their head. But I also want to come back and touch on the point about the virus mutating in an individual and the rate compared to billions of people, a couple of things to note there is, I think with any given strain, I mean, maybe billions is an exaggeration by a couple orders of magnitude as far as the resource pool available. But as far as I understand it, the theory on what happens in the individual with regard to like the emergence of Omicron was that you had immune compromised individual and multiple co-infections, and that there you had an exponentially increased rate due to the recombination with multiple strains co-infecting. And so I would like if anyone has expertise there to maybe address that point as well. Thanks.
I have a sort of a comment for he. So people have attempted to work out whether Omicron can be explained by recombination from, as you say, multiple infections, and I don't think that they came up with positive answers to that. Like the mutations that are present in Omicron, the ones that are, for example, coding mutations, they should be betrayed by silent mutations if they were really acquired from multiple infections of the same person. So you should be able to resolve the difference between the two. And I don't think that anybody has managed to do that properly. I think the experiment has been done. I couldn't put my finger on the paper, but I believe that has been done. And there's not an explanation. The idea that those patients had suffered from multiple infections at the same time, so they would be either infected by multiple people at the same time or sequentially infected by multiple people. That has not really been put forward, but people have tried to look for evidence of recombination as part of the origin and not really found it.
Okay, thanks. So I would be interested with those papers as well, but at least that's a plausible mechanism, and I'm not sure of the methodology for trying to replicate that potential, but it seems like that may be searching a vast computational space and of possibilities. Now, the silent mutation fingerprints, I understand your point there, and I think that's probably a stronger objection than the fact that they weren't able to reproduce the sort of mutations that they find. But yeah, I do think the lack of, again, I think I'd be interested to see those papers where the attempts were made. But yeah, with regard to the kind of zoomed out science level, societal level concerns regarding why can't investigators speak their actual minds, I think this is in some ways just as important as the origins question. But even on the origins question, we should all keep in mind that going back to the swine flu from '77 originating in a as a lab escape, or the anthrax lab escape from the Soviet lab as well, all the way up to more recent incidents with smallpox and so forth. That this is not just known, like as was mentioned with Dr. Ebright, and a lot of the discussion around the avian flu whenever gain of function was going on in 2012, 2014, that precipitated the moratorium. So it's a lot of people sounding alarms and a lot of kind of coincidences lining up that I think, at least for me, I think the balance of the evidence would lead one to presume the null hypothesis being a lab escape. But again, with the similarities with serial passaging, especially if you bring in chimeric introduction into serial passaging and the inability to kind of distinguish that from a natural spillover event, that question, is likely to never be definitively answered.
So, Heath, you actually brought up a good thing I want to end on. I want to transition to this real quick because I forgot. well, I now just remember that Sam has written a recent piece about something that I would say that everyone needs to pay attention to right now, which is the most important thing happening on this issue right now. And those are the events in Wisconsin. This law in Wisconsin has to pass. If it passes, it will be a huge deal. So if you've been following our Twitter account, you might notice that we've posted about a law in Wisconsin that would ban gain-of-function research, as well as require reporting of research with dangerous pathogens prior to any work with them. And it means that you would require scientists to report what pathogen is being worked on, where the work is being done, what is the work that's planned to be done, how it would impact the environment if it got out, all of these things, which would essentially transform science as we know it, because there no longer could be this veil of secrecy. Now you will say, well, it's just one state. What does it matter? It matters a lot, because that one state is the place where Kawaoka has a lab and performed those dangerous gain-of-function experiments back in 2011. And so Sam wrote an article about this, and I would say it's boring. It may seem boring, but that is where the fight is right now. That is exactly what, if any of you know somebody in Wisconsin or anything, if you could do anything to convince people to support that bill, do it. Because that's the tip of the spear right now. That will be a game changer for this issue. And that's why, in my mind, that's the most important thing we can be doing right now, is helping that law pass in Wisconsin. It will rattle people. It will rattle people. It will shake them to their foundation because of what it will hit. It will hit one of the top 3 labs in gain of function. So it's going to be met with like very, very big opposition. But if it passes, it will have a huge ripple effect, and you will see this occur in other states. And you might say, well, wait, what can we do if we're dealing with states? We really want a federal piece of legislation. I would say, consider this. If there's a federal piece of legislation, you only need to lobby against one bill. But if there's 50 states trying to do something, how thin are you going to spread the lobbyists? So I think that it's a sensible strategy. It's actually a better strategy for this to happen on a state-to-state level instead of a federal level, because it'll create the kind of chaos that's hard for the biodefense sector shills to stop. So anyway, Sam, I don't know if you wanted to say something about that, but I appreciate you wrote that article. And I think you've noticed that, as we have noticed, that this is a really critical thing to stop dangerous gain of function, but also give that transparency to the public that they just deserve as a fundamental human right.
You know, I wrote the article and I outlined, the sort of rap sheet of Wisconsin in this respect. I'm a bit ashamed to say, and I don't want to put Jonathan too much on the spot, but he used to be there, so I don't know if he had any additional thoughts, and I'm ashamed to say that I don't think I told him I was writing that article. And I might be doing a follow-up, actually, trying to get something into the Progressive, which is based in Wisconsin, so that progressives can sort of start getting awake to these issues. So I don't know, Jonathan, if you had any additional comments on that question.
No, just to thank everybody for their questions and comments, and Heath, if you want to get my I'll try to rattle my brains to work out what the paper was that I read to this effect. But if you'll have to contact me in order to get an answer from me, because I don't know really who you are. So thank you, everybody. I've enjoyed this conversation a lot.
All right, thanks, Jonathan. Take care, and I'm sorry about the technical issues. And when I post this on YouTube, I'll stitch it all together so it sounds seamless.
Yes, thank you, everyone.
And so remember, keep your eyes on Wisconsin, and we should be having another space pretty soon. I think we're hoping to host Chris Newby on one soon, but we'll also hopefully do one with lawmakers in Wisconsin to talk about that bill. So appreciate everyone's participation, and until then, take care.